Okay, hi everyone. We are back again with another biotechnology live stream. I am Danielle Snowflack, the Senior Director of Education at Edvotec. We're so happy to be able to bring you this training workshop today, again from the comfort of my basement. Hopefully I'm talking to you, somewhere equally as comfortable. We're going to do some science today. We're going to talk about epidemics, disease spread, and the ways that we can actually do a contact tracing simulation in our classroom or even at home. So thanks again for joining, and we're gonna have a great time today. So I know many of you have been here before, but today we are going to start by I just want to give you an introduction to Edvotec if you have not been to one of our live streams before. So we are the biotechnology education company. We were founded over 30 years ago by Dr. Jack Churchian, who was in biotechnology at Georgetown University. And what he found was that the exciting innovations that was happening in his lab weren't being translated into the educational space. So things like genetic engineering, recombinant DNA, protein production and purification, the polymerase chain reaction, all of these really fantastic techniques weren't being brought into the classroom. And, that led to, that observation led to the founding of Edvotec, where we want to be able to bring this innovation into classrooms. We really consider ourselves to focus on the education sphere in working with educators all over the world to help demystify science for their students and to really foster the next generation of scientific exploration. And so today we will be focusing on Edvotec kit S68, which is what is an epidemic and how does infection spread. So this is obviously a very relevant lesson right now, given what we are currently living through. But, I want to get away from just thinking about COVID-19 and thinking about other diseases that are common, like chickenpox or measles, or influenza, which is also coming up. So, all of these things are going to spread from person to person in a closed environment. And so let's talk about, what is an epidemic and what is infection and how does it spread? And so in humans, plants, and animals, infectious agents can spread from individual to individual, causing sickness. And even bacteria are not immune from infection. Small viruses called bacteriophage are going to actually seek out and attack bacteria, infecting them and using their cellular machinery to make more viruses. And so today we're obviously again hearing about, specific disease spread, but, the spread of disease within a community of any disease is problematic. So in today's lesson, we're going to talk about pathogens. We're going to talk about how they spread and how we can use testing and a little bit of detective work to determine the source of infection. And so first, I want to define a couple important terms. So one thing, what you'll see is in the chat window, we have a link to a survey. It's really more of just a form that will allow you to get the slides emailed to you afterwards if you would like them. We'll send them out really next week. If you would like a professional development certificate, we are also offering those for people who attend the workshop live. So if you would like a professional development certificate, please be sure to mark that you do want one, and we will send that out early next week. And then there's a comment section. If there's a time that's better for you, let me know the time or the topics for our next live stream. Okay, so back to the workshop. So I want to define a few important terms first. So the first three, which are outbreak, epidemic, and pandemic, are all kinds of facets of the same principle. And they all relate to the spread of disease within an area. But my understanding is that it's kind of like the difference in scale that's between outbreak, epidemic, and pandemic. Now, an outbreak is typically going to be the smallest of the three. It's going to refer to an increase in incidence of cases of a specific disease in an area that's in excess of the normal level of cases in the area. And so one place where we commonly will see an outbreak, and I feel like the cruise industry is not fond of this, but you'll often see an outbreak of the occurrence of norovirus, which is a stomach virus on a cruise ship. And it's problematic. If you're on that cruise, it's going to affect a large percentage of the people who are there. The normal incidence of norovirus should be 0. So if we're getting a lot of this virus is traveling through the community, it is going to be a problem. But it's restricted to that space and time. So it's a small, localized spread of disease. And so, it won't necessarily spread outside those bounds. An epidemic is one step larger. The pathogen is going to infect a larger number of people in a larger region. For example, due to a decrease in vaccination rates, there was an outbreak, an epidemic of measles in Samoa, in 2019 and 2018. Now, this affected a significant percentage of the population. Nearly 6,000 people were affected. And this, again, is because of their declining vaccination in the community due to misinformation about vaccines. A massive vaccination effort was able to slow the speed of the spread of the disease, but not before nearly 6,000 people got sick and 83 children died. And so, these numbers are problematic. But the disease did spread over a larger area. But again, it was largely restricted within the single country, over a period of time. Today, we find ourselves in the midst of a pandemic, and that's going to be the outbreak of a disease, a sustained outbreak of a disease in many different geographic regions, and that is going to affect a substantial number of people. And the thing to remember is that all of these definitions are relative, and they're relative to the disease state and the time of year in a particular area. And so one way we can think about this is the difference between seasonal flu and pandemic flu. I remember back in 2009, I did get a really nasty case of the flu, and my doctors thought it was H1N1, which was a swine flu that was going around. And so, the reason why the researchers became interested in this was because the flu numbers were increasing above what is normal. So, we know every year in the fall and winter, there is going to be a level of seasonal illness, and some of that is going to be caused by the influenza virus. And that is something to be expected as people get into closer quarters and move indoors for the colder weather, the dry heat. And there's a host of conditions that we know about in our environment, and even many that we don't, that could potentially increase your susceptibility to disease. But when we start to see the number of cases of flu increase above what is to be expected from historical data, that's when researchers and clinicians start to look at what's going on in the area more closely. And so this is what happened in 2009 when a new strain of the H1N1 swine flu emerged. The virus quickly spread through the community and caused more severe illness within the community because there was little immunity to the virus around. This wasn't a common strain of the flu, and so everybody was pretty susceptible to it. Now, to interrupt the spread of disease, public health professionals will identify and contact individuals who may have been exposed to a pathogen. And that's contact tracing. So when we start to really dig into a community through a strategy of testing and other medical interventions to trace back the source of the infection. And so in addition to allowing researchers to study the distribution patterns of disease spread, it allows clinicians to provide treatment and counseling to exposed people, to notify infected people so that they can shelter in home so they do not spread the disease further. And of course, this allows us to stop the spread of the disease within a community. All right, so we've talked about these basic definitions. So let's talk about different types of pathogens. So what is a pathogen? A pathogen, infectious disease outbreaks are caused by pathogens, which in the broadest sense is going to be any organism that can cause disease. And this is going to include viruses, bacteria, fungi, and even proteins called prions that cause proteins in your body to fold in the wrong way, which is going to affect your nervous system. And many of these exist in and on your body at any given time. But when conditions change, either in you or your environmental surroundings, the pathogens can kind of be activated, and they can invade the body and cause disease. Now, if anyone has any questions, please put them in the chat box, or anything you want to add. Please feel free to use that chat as a way to communicate with us. Okay, so we're talking about when things get out of balance, when conditions change within a person or an environment. We can get infection, and so that leads us to the epidemiological triad. And so there are many different models for the spread of disease. But one of the most common is this epidemic triad, where the host, the infectious agent, and the environment are all in balance with one another. And this is considered the normal state. So if you think about something like, strep throat, chances are you have these strep bacteria that are living on you and in your mouth and, in your body the entire time. But what happens is, and this is considered the normal state. Just because a bacteria is living on you or in you doesn't mean that it's going to be causing infection. And so, when this gets knocked out of balance, that's when we cause disease. So just to define some terms, in this model, the host is going to be the organism that is susceptible to the infectious agent. The infectious agent is our pathogen. The environment is going to bring the host and the agent together. And these 3 points of the triangle, so these 3 conditions are going to interact with one another. And so as long as they're in balance, we don't have a problem. Oops. I advanced one slide too far. And so as long as these are in balance, there's no problem. And we can kind of see that in the scale that you can see in the middle of the picture that I have on the slide. And when the balance tips, the host can become sick or the virus can be eradicated. And so there are examples of how that balance can shift on this slide. So if a virus becomes more pathogenic, so that means it can cause more severe sickness, or it's easier to spread, it's easier to infect people, the scale is going to be tipped in favor of the agent, and the individual will get sick and be able to spread disease throughout the community. If the numbers of the pathogen changes, the scale can be tipped in favor of the agent. So we could think about, right now with respiratory viruses, and this is being indoors versus outdoors. So if you're outside breathing with a lot of people, there's a lot of air around us, and you get a lot of dilution of the virus particles and respiratory droplets. But when we go inside, the air is not moving as much, there isn't as good of ventilation. And so instead of being exposed to 10 infectious particles like you might be outside, you're going to be exposed to a lot more. You might be exposed to 100 virus particles by being inside where the air isn't moving as well. And so, we can also tip it in the favor of the host. So with a vaccine, the number of hosts that are able to be infected by the pathogen decrease. And, that would result in fewer people being infected. And furthermore, the environment can change, For example, we see cough and cold more often in the fall and winter when we move indoors. And so to best control the spread of disease, all 3 of these factors must be considered. All right, so there are many ways that disease can spread between people. And this graphic, does a great job of outlining them all. And so I linked to the article that this was pulled from. So if you do request the slides by completing that link, you'll be able to actually go to the article, the little write-up that this came from, which was a nice little write-up from a few years ago. But this picture I just felt like did a really great job showing about how disease can spread between people. So pathogens can enter the body through abiotic or physical means. This could be the wind-spreading fungi, spores, or entry of parasites into the bloodstream through cuts in the skin. Animals are also common vectors for disease. So one of the vector is an agent that carries and transmits a infectious agent, a pathogen between human organisms. And one common vector for this is the mosquito, which can transmit malaria, yellow virus, or yellow fever and Zika virus, combining the two there. But, and the mosquitoes are actually responsible for transmitting other viruses. So, they are pretty nasty little bugs. And so, those are some modes of transmission. And viruses are often transmitted between humans. And there's a lot of ways, just by living our normal life that we are going to transmit viruses between individuals. So we can transmit disease directly from human to human transmission. And a lot of times this comes through fluid exchange. by whichever means you would like to would like to discuss with your students. So blood transfusions, etc. Intravenous drug use. And so this can be in contact through direct contact with body or blood or bodily fluids from infected people. And that's like Ebola, which is transmitted through fluids. And, it's a little bit more difficult to get these because you do need the direct contact with the fluid to get the disease. Many times these diseases are easier to kill because they need to be within the body's fluids and they don't last long when they're outside of their preferred environment. Indirect contact happens when an infectious agent can actually survive on other surfaces for long periods of time. And you can actually pick them up from contaminated surfaces. And you might hear about people talking about fomite transmission, and that refers to transmission from objects that are likely to cause the infection. And so that's like doorknobs or clothing, or even our favorite omnipresent cell phones. Cell phones are actually covered in germs, and so that's a good idea to keep your cell phone clean. Because these are the way that, viruses like the flu or norovirus are easily transmitted between individuals through this indirect contact. Now, droplet or airborne transmission, so they both kind of refer to liquid droplets that are going to come from the mouth, the respiratory sinuses, that are going to go into the air when we cough or sneeze or talk. And so the largest particles don't actually last long. Those are the droplets. They're heavy, they fall out of the air. But they do contain viral particles, and so this would be an instance of if I were to sneeze on you and you were to get those respiratory droplets on your face, the virus or bacteria could get into your body through your nose, mouth, or eyes. Eyes are actually a really easy place to have an infection get into your body. Now, aerosols, the smaller droplets are the aerosols, and those can actually travel through a further distance to infect people. And so these are common modes for respiratory viruses. And again, there's one more route, the fecal-oral route, which, they all sound pretty gross, but this one sounds particularly gross. And so this is where pathogens in human waste enter the food or water supply through contaminated water or poor hand washing after using the bathroom, lack of sanitary conditions. And so this is an incredibly serious public health issue because there are a lot of waterborne illnesses that can happen when sanitation is not sufficient. So a common pathogen that was spread this way is cholera. And so with modern sanitation and water treatment, cholera has largely been eliminated from, industrialized areas. But in parts of the world where they don't have the same access to clean water, it's still going to affect millions of people per year. And so, this is still a serious public health concern, even if, people in industrialized areas don't necessarily consider cholera to be a disease, to be a problematic anymore. All right, so let's go to the next slide. So these are a couple ways, a couple protective methods that our body uses. Like thinking about our last diagram, it seems like there's a lot of ways to get sick. And, we're constantly bombarded by infectious agents. So why aren't we always sick? And sometimes it feels like we are, but, luckily the human body has some proactive methods that will be able to help us from getting disease. And so luckily we have these methods or we'd always be sick. And so the first is simply a physical barrier. So, we just prevent the bacteria or the virus or the fungi or whatever from getting in to start. And so this is going to include our skin barrier, which is largely impermeable. It's going to keep water and our body fluids in and keep, bacteria and fungus and other bad stuff from getting into our bodies. So the water stays in, the bacteria and pathogens can't get in, they stay out, and we prevent infection that way, just basically do not enter sign. Next we have the innate immunity. And so this is the body's response to just general pathogens, just general problems that arise. So if you were to get a cut, we were to breach that barrier, the first thing your body would do is kind of flood that area with immune cells. We're going to get platelets there to close up the wound, but we're also going to get white blood cells and a variety of white blood cells that will get in there and start targeting anything that's potentially infectious. And so that's why we're going to have the swelling, the redness, the warmness at the site of these injuries, because that's your immune system starting to work. Now, after the innate immunity, we start to get to the adaptive immunity. And so this takes a little time to build. So because your immune system is actually creating a specific response to the pathogen. So we're targeting it specifically with these, highly specialized molecules that will target pathogens for destruction by the immune system. And so in the first reinfection, you might actually have a serious infection because your body doesn't have the antibodies. It doesn't have the tools yet to quickly shut down that infection. Once your body does create those antibodies, it's able to neutralize the pathogens. And the thing about the adaptive immunity system is many times, the majority of the times, the body remembers that pathogen. If the pathogen enters the body again, the body already has the tools it needs to, attack that virus or that bacteria more quickly. And so in many cases, the host might not even get sick or the symptoms would be mild. And the nice thing is, if you want to study the immune system more closely, we offer our model, this origami organelle, where your students can study the immune system more in depth, even if you're remote. So this is an option for your students to actually try, doing some antigen-antibody interactions and some response to disease. And so researchers are going to evaluate the spread of disease using the R0, which is the average of how many people will contract a pathogen from one infected person during their infectious period. So you might have heard this, I feel like we hear this number in the news a lot. It does seem to be changing, but this is, science that is happening right now, in that we're learning more and more about what is happening with the SARS-CoV-2 COVID-19 epidemic. And so there are many variables that do factor into this number, but the three most important are going to be the number of people the infected person contacts, the likelihood of infecting a secondary contact, and the infectious period. So if one person had contact with 3 people per day and had a 1 in 3 chance of being infected, and the infectious period was 3 days, so the time you were being able to shed the virus and actively infect people around you was 3 days, you'd have an R0 of 3. In general, an R0 above 1 means that the virus is able to spread quickly through a community unchecked, and the number of people who are infected will increase exponentially, increase quickly. An R0 below 1 generally means the spread of the virus is the pathogen is low in an area, it's slowing and it can be contained. And so the important thing to know about the R0 is that it can actually change over time. And the numbers we have here for the avian flu, the plague, Ebola, those are averages based on past outbreaks. And what I do like to point out is that if we look here at some of these diseases, the flu or Ebola, or even SARS, the initial coronavirus that caused a pandemic, for 15 to 18 years ago, I forget exactly when, there are not levels are, 3 to 1.3. So they're on the lower side and it's easier to contain them in a community. But if you look at the R0 of measles, it's 16. And this is based, again, on historical data, but it just shows you how infectious measles is. And we do think of it as, a disease that, isn't a bad disease. But if you look at how infectious it is, it can spread really quickly through a community. It can, really affect children who may be immunocompromised. And while, measles may not be fatal, there are a lot of conditions that can be caused during measles infection that end up resulting in chronic conditions. So, we think of measles as being a common childhood illness, but it really problems that could affect a person throughout their life. And again, it's one of the reasons it's an incredibly effective and safe vaccine. And this is, one of the reasons why, the vaccines do help the disease from spreading. And so, but for current transmission, what we're seeing right now with SARS-CoV-2, which is a new disease, it's a lot harder. So the R0 is constantly changing and it can be different within communities. There's no historical data, but it can help scientists, doctors, and public health professionals make recommendations for policies to help contain the disease locally and regionally. All right. So, the one thing with infection is that prevention is always the best medicine. If you don't get a disease, you don't have to be cured from a disease. And luckily, with the proper precautions, we can prevent the spread of disease. Airborne pathogens like COVID-19 or the flu spread person to person through liquid droplets that come out when you cough or sneeze, these respiratory droplets. And so that's why when you sneeze, it's better to sneeze into your shoulder or sneeze into a tissue, to kind of constrain those respiratory droplets. Furthermore, we can wear cloth masks to cover the mouth and nose, which prevent our respiratory drops from spreading. And right now is able to spread through these means. We can take actions like social distancing to reduce the likelihood of infecting those around us. Gastrointestinal diseases will spread through contaminated food or infected surfaces. So this was back like that norovirus, which is a gastrointestinal disease. Foodborne illnesses can be prevented if we are careful and use proper food storage and handling techniques. We make sure to keep common areas where people might be eating sanitary, and we make sure that food is stored, at the correct temperature to prevent bacterial or, the growth of other agents. And so, and one another thing, it's really important to practice good hand washing because, this is a simple step that can help us really cut down on the number of disease, especially in the winter when we're all in close quarters. Because again, touching your face with contaminated hands can introduce a virus to your mucous membranes, to your eyes, and to your mouth. And so it's really, really important to keep your hands away from your face. And of course, you see me touching my hands with my face a million times, and we all do it unconsciously. But if you keep your hands clean, you're reducing that risk. Luckily, soap, hand sanitizer, and other disinfectants can kill most pathogens. Again, so wash your hands, keep your hands clean to limit the spread. If you're exhibiting serious symptoms of anything, make sure to contact your healthcare professional for guidance. And they'll be able to give you the information. The best source of information is going to come from your doctors, the CDC, or the World Health Organization. Okay, so we do have a question. What is the longevity of the virulence of COVID on a surface? So I don't have the exact numbers. I didn't look them up before, before we started this. I do remember in the beginning, it was a big problem, this fomite transmission, and people were really worried that this was going to be a major mode of transmission. I do think that current research has shown that it's less stable on surfaces than we had initially thought. I feel, and so this would have to be substantiated. And so I can look up that if you fill out the form, I can look up the information and get it to you after the workshop. But I believe that I had seen that it's, it retains its virulence for 24 hours or less once it's on a surface. So a day is still a long time, but it's not, incredibly stable on on surfaces. And again, the viability of an infectious agent on a surface is going to be dependent on that surface as well. So some, hard surfaces, versus soft surfaces, porous versus non-porous. You could imagine something that wicks away water would kill, a bacteria pretty easily. And so there are a lot of factors involved with how long, any infectious agent, let alone COVID, could particularly live on a surface. I hope that answered your question. And again, please, if you submit the form, put the note there and, we can chat more about that. Okay, so we are going to perform a simple experiment that's going to allow us to chart the spread of a disease through a community. So let me just go to my setup. Let me get my camera started. And we will talk about the experiment. So we are going to be doing disease transmission in a test tube. And so this is going to be kind of like modeling what would happen, at a party. And so, this would be a short time with a moderately, infectious disease. And people are going to be infected, but they're not necessarily going to have time for that virus to start shedding within them, and then in turn, then for the virus to start replicating inside of them, and in turn become infectious themselves. So this is what could happen if we were at a party. And so I basically performed all of these steps already because I could imagine that sitting here and watching me pipette liquids into tubes would be incredibly boring. But, I just want to kind of like run through what I did in terms of the protocol. And then then we'll actually go and start doing now analyzing the results. So for this protocol, each student would get a tube and you would probably number them to put a personal touch in. I named them all. So, we've got different names. Here's Gail, and so each of these tubes would represent a different student. They would actually remove 1 ml of sample from their tube. So we would remove it into a test tube. And we're going to have 3 contacts, so they would remove 1 ml from their tube, and they would do this 3 times. So we're just going to put it in a microcentrifuge tube. And your students would randomly choose another student in their classroom to exchange fluids with. And so we can imagine this is a handshake, this is sharing a drink, any means through which you may be exchanging fluids. And we would give this to the other student. Each student would then open the tube, they would remove the liquid and then put it back into their tube. And now we would repeat that 2 more times. So it's going to be 3 times in total. And so again, we are removing all of the sample, all 3 of the samples into microcentrifuge tubes before joining the party. So this is going to be, we're only going to be able to mark the spread of who came with the disease, right? And so, one person has the disease, they contact people, that virus isn't able to replicate in their body in the short time that they're all together. And so your student gets their 3 tubes, they empty their 3 tubes into their 1 tube, cap the tube and, flick and invert to mix because we want to mix the samples from all 3 of the students' contacts plus their own sample together in their tube. And then we are going to add our indicator dye to these tubes. And so again, each student will have 1 tube that's theirs. You as the teacher should definitely keep track of which ID number or which name, gets each tube, because one of these tubes is filled with a solution that we're going to be calling our pathogen. And so it is just, in this case, it's a base and an acid, and we're using an acid-base indicator that's included with the kit, S68. And this will allow us to see a color change reaction, which will suggest that our students came in contact with someone who was infected. And so I'm going to add 1 drop of indicator to all of these. And the people who turn pinkish red are going to be the ones that were exposed to the disease. And so what you can see is I get this rapid color change where you can very clearly see the differences between individuals who have been infected by our simulated infectious agent and the ones who have not. Okay, and so we can see, ideally you would mix these up a little better. I don't want to cap every tube and mix them. But what I'm going to do is cap 2 of them and mix them so you can see the color. I mean, you can very dramatically see the color change. But, I just want to show you the difference between Hank, who is red, who is clearly infected, and Brock, who is white, who is yellow, and is clearly not infected. And so your students would have these clear solutions. They would walk around the class exchanging fluids with one another, and then we would do this, essentially a test. And so these tests allow us to determine who was exposed to the infectious agent. In reality, many tests wouldn't be able to give us positive results right after transmission. You'd have to wait a couple days for that pathogen to get into your body and be able to reproduce. But again, this kind of would give us this essentially would give us a snapshot of what it would look like a couple days after this party. And so, we repeated these steps. At this point, once we have the results, your students are going to identify, who is infected. And so here we can see Hank, Bryn, Anne, and Michelle were infected. And these names were picked at random. I did I picked these names at random, so it doesn't, there's nothing really noted here, but you can see here that here are the results from our testing. And so how do we go from here, and how do we look at this pattern and determine who was the person who was first sick at the party? And so this is where contact tracing comes into place. We're going to kind of do some viral, some pathogenic detective work. So we know who's positive. We know they were all in the same party at the same place. And we can kind of try and figure out who contacted who. And so I have this chart. You would make a chart like this with your students. Again, to keep track of everybody that they contacted over the period of time. And in our results slide, you can see people who are positive are red, and the people who are negative are green, their names. And then to identify the source of infection, we need to analyze the contacts all of our individuals at the party made, but who are most particularly our infected, individuals made. So I'm going to color all the people who were tested positive in red through our 3 rounds of transmission through infection. And what we can see is that when we look at the pattern, there was one person who was in contact with all of the infected patients, and that's going to be Hank. And he is the person who was in common with all 4 infected people. And, that's just from, saying, okay, at the end, we knew these 4 people were sick. How were they related with one another? And how can we use that to show us something? And so, again, if we were to kind of reorganize this information and put it in a flow chart, what we can see is that Hank was in contact with all 3 of these individuals. And so he would be, our patient 0, the person that the infection did start with. And so now we have identified who in the community is sick, and we can put protocols in place to limit any further spread. Model, our people didn't get infectious right away, and so the disease didn't spread that rapidly. It didn't spread through so many people. But what would happen if we were in a community and we gave infected people a day or 2 to become infectious before making their next contact? Where the people are together every couple days, and one of them is sick. We have these interactions. We go away for a few days. We come back. It's enough time for that virus, that pathogen, that bacteria to really get into the body and to start reproducing. And so we can model this by mixing the patient samples during the experiment instead of after the transmission. And so what we would do is instead of, let me turn my samples around. All right. And so what we would do here would be instead of removing our samples into separate tubes, before performing the experiment and making the exchange of fluids through the tubes, what we would do is remove the sample right from here. We would place it in another tube. And I know this is how this is going to work out. So, we would do this, we would remove this, and we would just exchange the fluids directly. And so that's really going to model what would happen if, a person was in contact with an infected person, and that virus, that bacteria was allowed to have time to reproduce within the human, and that human was able to become infectious. And so then we add the indicator dye again, just like we did before. Only again, these people were making contacts, going away for a few days, becoming infectious, and then being able to interact with people again. All right, and so what we have here, I'm going to go through again, and I'm going to add my indicator. And so what you can see is that we are getting a lot more people being infected in our simulation. Okay, so if we look here, we only see that we have Mike here and we have Skylar here. They're the only 2 people who did not turn pink, who did not get infected with our, infectious agent. Again, if we go back here, we see that there were only 4 people who became infected with our infectious agent. And so you can imagine this is going to make it a lot more difficult for our contact tracing. So how could we even start? How could we try to trace this? And so the way that I usually do is that I start to look at the healthy people to try and analyze the spread of the disease. And so here is our slide, again, the chart of all of our interactions, and then the results. So you can, Skylar did remain by the virus, and the rest of the students tested positive. And so since they tested negative, that we know that any contacts with them were not responsible for passing the virus. Furthermore, Skylar and Mike did interact with each other in the third round, and neither of them tested positive. So our confidence can be high that they were not infected. And so we can eliminate them from being our patient zero, our first sick person. And so since we know, so I'm going to color them in their names in green throughout this whole chart. Since we know that anyone who interacted with Skylar and Mike were also negative before and at the time of their interaction, they weren't necessarily sick at that time. And so, we can rule out those people who made contact as well. So I color their names blue here. And so we can, hold on one second. Okay. And so, please let me know in the chat window if you're having problems with the video. I did just get a note from YouTube saying that there might be a problem, but it does look like we're still transmitting. So, please let me know if you are having problems in the chat box. Okay, so since we know that anyone who interacted with Skylar and Mike were negative both before and at the time of interaction, I color their names blue here. And so we can actually also rule out people who made contact with people we know were in contact with people who we could figure we knew were negative. And so in this case, we can rule out Hector in the first round. We knew he was negative in the first round because he was able to, oh, good. Thank you for letting me know. I just got a message letting me know it's cutting out a little bit, but not so bad. So please let me know if you miss anything, and I will go back to cover it again. Okay, and so again, we know that Lydia interacted with Skylar in the second round. We know she couldn't have been infected in the second round because she was able to interact with Skylar, and Skylar never tested positive. And so through that, we can infer that Hector was not infected in our first round of transmission. And again, since he was not affected in that first round of transmission, through process of elimination, we can also say that Lydia was likely not infected at that time. And so we kind of go through and make those connections and make those inferences based on the interactions between the people. And we can trace back here, in this case, to two different individuals. And so what we found here is in round one, it's likely that Holly and Saul were infected with the disease. And so, we can chart the spread of the disease through the community in a flow chart again like this. And so what you can see here is that, Holly and Saul had the interaction in the first round. In the second round, they spread it to each to another person. And in the third round, each was able to transmit it to still another person. And so you can imagine ways that, had Skylar and Mike not interacted in the last round, everyone would have tested positive. And that could be problematic. What you can see here is that we can't eliminate either Holly or Saul. We don't know who patient zero was. And that's the person who had the disease first. Our cross-checking and our disease detective work allowed us to trace back, but we can't entirely rule out someone. And so I set up the experiment. So I know the person who was sick was Holly in the first round. And she was the one who then, through her interactions over time, was able to infect the other individuals. And something that's important from this lesson is that it serves to demonstrate that there may be some instances where there's not a clear conclusion. And so today, if we relate this back to the current pandemic, this could happen in an area where there's really high levels of community transmission, where we can never rule out an interaction, where there aren't enough, if we're in places where the percent positive rate is perhaps around 20%, it's really hard to contact trace because we can't find enough people, we can't find enough connections to figure out where it came from. Now, when community spread is low, that makes it easier because the diseases form in clusters, and then we can rule things out from there. But this is actually really important because ambiguous results like these will allow you to start a discussion with your students about the challenges facing public health officials when contact tracing and how hard it can be to identify the source of an infection. And this is important, because these are real world problems that your students may be seeing every day. And so now, where do we go with this activity? And so there are actually a lot of places that we can take this super simple experiment through some extension activities. And so first, using this system, so this, this acid base indicator system, we can actually model different problems and different challenges and different treatments within a community. And so one thing that you can do is develop a system to model the interaction of a vaccine or herd immunity. And so this could be between buffering the solution, changing the components of the solution. Some students might get something different in the beginning. And this would not allow our virus, our infectious agent, to spread between individuals. And so that way we can, this, is a vaccine or herd immunity type model. And we can actually also model social distancing or mask wearing. And so one thing you do is that, one student's got to stay 6 feet away from everybody. And if they never get close to another student to be able to make that fluid exchange, the student can never get infected. And so that would be a way to show how social distancing would actually work in the context of disease. One fun extension that I saw online was actually to model doing a nasopharyngeal swab. And so that's what you would do for the flu test or the COVID test, where a clinician would take a big long swab and put it all the way back into your sinuses to sample from that fluid. And so through sampling that, you'll sample that fluid much like you would do with a flu test. It's obviously going to be on a lot larger swab than this one, but we're going to use this as a model. So I am going to swab patient A first. And so again, we're going to use this vessel. This is going to be our model for our sinuses. So we can imagine this is a respiratory fluid, mucus, etc. We're going to take a second swab and swab patient B with our Q-tip. And then we're going to add the indicator dye here. All right. So let's go here. Let's see, can you see this okay? I'm just going to add the drop of dye right there. And what you can see is that again, you can see we get the rapid color change, showing us the difference between a positive test and a negative test. So again, the yellow is going to be negative for our infectious agent. The bright red pink is positive for our infectious agent. And that is kind of, models the nasopharyngeal swab. So, the way that we actually get the respiratory fluids to be able to look for the presence of viruses. And so that's just kind of like a fun, I don't know that this necessarily, changes the science, but it's kind of fun because your students are actually modeling the medical test as well. So I just think that's kind of neat. One last thing is if you're not in class, you can actually use a random team generator. I linked one on this slide. So you can have your students try 10 students versus 50 students. Again, in groups of 2, have them go through multiple rounds, of, connections. And then, you can assign your student, a random person who is either the person who's infected, and then they track the virus spread through that population. So if you're in the lab, there are further extension activities you can do. And one thing that would be cool was to actually pair this model with one of our testing simulations, which would allow you to both talk about the medical test, the disease, and its spread. And we have multiple disease detection kits that would work really well with this. We have a flu detection kit. We have a COVID ELISA. We have one that is a simulation of an Epstein-Barr disease testing kit, which is going to be the disease that causes mono, which can be common amongst students. And then there's also an HIV detection ELISA. And so, we compare these testing simulations with the simulation of spread and really have a comprehensive lesson on how diseases spread and the importance of testing in determining these problems, in determining these diseases, and then in strategizing on how to prevent them. So you can have your students get together and brainstorm what they would do to prevent the spread of disease and how we could put it into place. And so just to kind of wrap up, if you have any questions, I would put them in the chat box now since we seem to be having a little bit of lag. I want to make sure I see them before I wrap up. But, I'd love to answer any questions you might have. So just to summarize, in this, workshop, we talked about how we detect and analyze the spread of disease. Our immune system is a big way that we can prevent disease. So the human body over time has evolved protective measures to protect us from disease. This comes from the impermeable barrier, that is our skin, to our immune system, both the innate and the adaptive immune system. So the way our body responds to any pathogenic agents versus ones that it has been exposed to previously. And the biggest thing is that disease, we do see that individuals become sick when the balance between the host, the agent, and the environment are tilted out of balance. And so that's when disease will occur. And that could be something, again, like the move from outdoors over the summer to the indoors as weather gets colder. And we can see, currently during the COVID-19 pandemic, we can see as we move inside, the transmission of disease happens more often. And simple experiments like this. So this was just, allowing us to exchange fluids between students and analyzing the results of the color change reaction. Simple experiments like this will allow us to model many different principles in public health. It'll allow our students to learn the importance of testing and having many people tested, and the methods through which a disease can spread through the community. And so, again, as I mentioned, we will be posting the presentation and the slides over the next few days to our website. If you want me to email them to you when they are available, please fill out the form that Maria posted to the chat, and we will email them to you within the next few days. I just want to say thank you so much for your time. It's, really great to be able to run through these experiments with you, to have you be able to play along with me and see the results. If you have any questions about this experiment, you want to try and figure out how to work it in your classroom, please get in touch with us. The best way to contact us is by phone or at info@edvotec.com, or try one of our social media channels. We really look forward to helping you get more biotechnology into your classroom. And again, given the current events, this is a great way to get your students engaged with the material so they can kind of start to better understand what is happening around them with the COVID-19 pandemic, and to start, strategizing about ways to prevent it, and, even start thinking about, therapeutics, treatment. It's really the jumping off point for discussion of a lot of areas in human health and well-being. And so with that, if there aren't any questions, I just want to say thank you so much for spending this time with me. And I look forward to seeing you in a few weeks at our next live stream. So thanks again. Wash your hands, stay safe, and we'll talk soon.